I did rework my pictures, and I got what I think are better pictures, and I rearranged them all. Made it work. So let's go ahead and undo it. At this point, you probably understand what I'm getting at when I talk about recessive and dominant. And, you know, I didn't know where to spell them ear lobes, as in two words. But everywhere I looked, it's earlobes. And so I also made it earlobes. And it's like gallbladder, really is gallbladder. Urinary bladder is two words, and I've learned that. So that word free, I've seen it used, I've seen detached used, I've seen unattached used, and I saw free used more. So free would be hanging down like this, it's not attached. And I reworked all these pictures to make it better. I still like a lady with the earring and a man without. That's just me, old-fashioned chauvinist guy, I guess. But anyway, I try to have them facing each other, which I had time to fix that out. All right, now 32. To be free like that, as you know, there are two ways you can be free. You can be two big Fs, which is homozygous dominant. Or you can be just one F, and both of them will look the same way. You can't tell by looking what they are. But that lady there would be free, unattached. And it could be either the homozygous F's dominant or the heterozygous. So that was what I get across to you. And the next one here, there's just one way that person can be attached. That's the recessive trait. It's got to be two small F's. That's all there is to it. But the big F would make it dangle. Okay? So that's, you know, there's two genotypes for the dominant phenotype, and there's one genotype for the recessive phenotype. And those words confuse me sometimes, trying to say them all together. Okay? The next one, now, I'm going to throw a question at you. What are the odds that a person which is homozygous recessive, free, that'd be this person. And I tried to match them though. The first person is over here. And a person who's like this would have children with the free earlobes. And how about what are the odds for this way? Now, I made my Punnett square. And this person are the two big F's because, like I told you, homozygous free. And this person's right here. And that'd be the two small F's. And then when I recombine them, I find that as far as producing a cubic free earlobes, every time. No chance to make a child with attached. Because every time that big F is going to stop it. It's going to be free. Yes, ma'am?
That's homozygous recessive? The little letter earlobe?
Yes.
Okay, so.
Yes. Homozygous means the same, and recessive means the little letters. What you do not do is use an F and an A. A lot of folks want to do that. But you want to use the same letter, and just a smaller one is another way. And then I said, let's get another way. What are the odds that a person who is heterozygous free, and all I did was change this parent. This parent is homozygous free. And I said, what if they're heterozygous? So I'm doing heterozygous free. And of course, we know what that person is. That's two small letters. So when I make my Punnett square, now I got these two alleles because this person is big and small. And again, this person can only produce these. And now when I cross them up, I find that's about 50-50. Out of 4 young ones, 2 will be free and 2 will be attached. And that's why I said 50-50 chance. So if she had 10 children, it might be 4 and it might be 6. It's still, unless, huh?
Look at your Punnett square.
My Punnett square.
The bottom, yeah.
Yeah, this should have been over here. I believe I can quickly fix that. It works. Oh, I want this to come off first. I want to take this and drag it up, put it right there. Then I want to play it and make sure it works right. I want to show this first to you. So now, after I show the square, then I can show you what the odds are. And then the next one was if you have two parents and they're both heterozygous free, what are the odds of having a kid with the free earlobes like both parents do? And what are the odds of having a kid with attached like neither parent does? That business divorce causes that too. Where the child is born that don't like it, and they think they cheated on you. Well, the wife knows she didn't, and the husband should trust her, but some guys are stupid. So in this case, unless I screwed this up, I didn't. This parent would be these two alleles. This parent, same two alleles, and then when they join up again, I will find that 3 of them out of 4 will be free. And that should be a 75 right there. And 1 out of 4, that should be a 25, and there they are. I don't guarantee it, does it? Because you can have 2 kids, and both of them by chance might be this. Don't mean the postman came by, that means that you both heterozygous. Okay? So that's how that works. Sickle cell anemia is one that is near and dear to many people. And then do you realize that blacks are the ones that have no chance of getting it either? A lot of the countries in the Middle East have high percentages of sickle cell. In our country, it is the black citizens that have the most, but that's not worldwide. And I see right here, I need to take it. Oh, I won't do that. It's too much to fix. I was going to put a red on this thing here, but I don't want to do all that messing right now. But anyway, sickle cell anemia is caused by a recessive trait. You got to have 2 small genes to have the disease. The hemoglobin, HB is what we use to stand for hemoglobin. Hemoglobin A is the one that is all right. That's the one you okay. Hemoglobin S is the one that gives you sickle cell. Now this time they do use different letters, and that's just the way they've done it. You know, they being the scientific community who did it before I did it. I would have used probably a small a in there, but they chose not to. But these are real photographs taken through a scope, and you can see that this is the shape the cell's supposed to be in. These are sickle cells. And they do not roll. They will flip-flop and cause a blockage, and then what's below there doesn't get fed, and the pain is intense. Transfusions are done a whole lot to remove the bad blood and put back in the good blood. There's another picture showing the sickle cell. These are okay. And I understand that when the person who is anemic, when they make their cells the first time, they all look like this. They all look good. And then that cell goes to your lungs and it picks up oxygen. That's good. And that cell goes to your tissues and gives away the oxygen. When it gives away the oxygen, it then turns to this shape. It carries at one time. And upon giving it up, because of the bad hemoglobin, it takes on a curve, a comma shape. Well, these things are supposed to roll like you might roll a quarter across the floor. You can't roll a cracker because it's not round anymore. So, and I found me even a better picture, which is through a microscope. Can y'all find the sickle cells among that sample? Do you see them? Do you think there are a whole lot of them? But this cell, good or bad?
Good.
That cell.
Good.
That cell.
Good.
So you can see them. You can see them. What do you think those things are? No, those are white blood
Those
cells.
white ones.
They're white blood cells. Why are they white? Because we stained them when they die. And the nucleus sucks it up. Why are there no big blue marks in these red cells?
No nucleus?
They don't have a nucleus. You see that sunken in place right there? That's where a nucleus used to be. It's gone, so it sinks in. Out here, is pretty thick out here, thin in the middle. And the reason is because these nuclei, they love to eat oxygen. They love oxygen. Well, if this cell's job is carry oxygen, and he had a nucleus, when he got where he's going, the nucleus was going, that was good. He had nothing to give away. About like me cooking cookies for the church. I'll cook a dozen, I'll give them one. I eat 11. I would be a very bad nucleus, very bad, very blessing. But I couldn't, you know, these are nice, but this one, when I saw it, I went, oh, that's like doing our scopes. And beautiful. I often wonder what would happen if you did like a class of students and you did a blood sample and one of the kids had this come up. I would suspect they know that they're anemic already. But this truly is and you see where the field is. I don't like that blur. When I do my camera, I make that nice crystal clear. Okay, so that's a picture of sickle cell. Now, these are the way they look. This person is healthy. All their cells look like this. This person's a carrier. They're healthy. All their cells look like that too. That's a piece of a round cell. There's no problem here. But this person, there's no way they can be this way because all they have are the bad alleles. They're going to be sickle cell. And this is the way, these are two alleles. This is homozygous, okay. Heterozygous, okay. Homozygous, sick. Now, between this one and this one, did you know this one has an advantage over that one? Do you know that? This one right here, if you're this, you have an advantage over that person. Do you know what it is? You will never get malaria. If you have, and we don't know if that might be the reason you had it in the first place, if you have one of the bad alleles, for some reason, and I don't know why it is, this person will not get malaria. This person would. This person, I don't know about this person. I reckon they have sickle. That's probably worse than malaria anyway. Now, if these two people marry each other, they could live in Panama and never be fear of getting malaria. But they have a youngin'. Could that youngin' have sickle cell? Sure. If this came be, and then the rest of my slideshow is about that. What are the odds that a person with sickle cell anemia and a homozygous healthy person would make a child with sickle cell, and the odds are without sickle cell? Now this person is homozygous healthy. So if you look at the possible, this right there are their two alleles. There's their two alleles. Do you see any sickle cell kids in that bunch? Not one. Because this parent will always trump this parent. Now,
well, let me ask you, because the last question you were asking, one parent had sickle cell, the other one didn't. Wouldn't that be the dominant gene that did not?
but if
And
you
it would always
didn't,
overrule it?
but if you didn't, and you were this, you see, you can be didn't and be this, and be didn't and be that. Neither one has sickle cell.
Right. You said if they had a kid, the middle one.
Okay, if this person and this person had a kid, these two people had a kid,
Then they would never.
every time you had a match, this will always trump this.
Right. Oh, you're saying the middle one and the bottom one had a kid.
I'm saying
Okay.
this one and this one, not the middle one. I'm saying this one had a kid, but this one.
Then it would never be.
So this spouse would have sickle cell, but the kids
Would not.
would never have sickle cell.
Right.
Now what happens if these two get together?
Then that's when you had the chance.
That's your chance.
I didn't see what you were saying.
Okay, so this person here is sickle cell. Therefore, I know they are the HBS. I know they are. And this person I said is homozygous healthy. Homozygous healthy is going to be this. And when I put the Punnett square together, this one, there's your homozygous healthy. And there's your sick one. And every time they join, that A will always cancel the S. So all those kids. But now on the last one now, without sickle cell, all of them. Person with sickle cell,
So both of them wouldn't have?
none of their kids will have sickle cell. Because look at them. They all got the big A. They're all carriers, but none of them have the disease. And all of them maybe would be protected from malaria too. And now, if this person married a person like them, then they have a chance of having a baby with sickle cell. Okay? Let's talk about this one. What are the odds of a person who has sickle cell marries a person who is heterozygous? They're healthy, but they have the bad gene. Could they produce children with the disease? And what are the odds to have a kid? 50%, 25%, what are the odds? Well, now here's now there's this parent again, all they have is, this one this time is one each. I did say they're heterozygous, right? Now,
1 and 4. 1 and
so what are the odds?
1 to 3. Is that right?
Look at it again, Roger. 50-50. AS, SS, AS, 2 out of 4, 2 out of 4, 50-50 like Katrina said. Now what I did not do, and I think I'll do it real fast, is healthy. Ooh. Healthy. Will produce a child with sickle cell. And then I show you my Punnett square, and I explain to the class that for both parents, because they're carriers, they're going to have this breakdown, this segregation. And then when these two get together, this person's okay. These two, that'd be okay. These two, that'd be okay. There's your combination. 1 chance in 4, which is 25%. 3 chances in 4, healthy. Now, let me ask you another question while I'm thinking about it. I want to slide this up higher. So folks in the back will see it. Now, I've got another question. So I'm going to add down here at the bottom. Many, I can spell, are homozygous healthy. Okay. How many are heterozygous healthy? And how many are sickle cell? No, I'm going to change that. I'm going to keep using my same format. Homozygous recessive, which would be sickle cell.
Is heterozygous recessive the same thing as a carrier?
No. Oh, yes, yeah. But there's no recessive then. He's just plain heterozygous.
Okay.
If anything, you might say dominant, but that to me it means dominant. So, all right, now. All right, now. If you think back to your square, of those 4, how many of them was A and A?
1?
How many were A and A?
1.
What percent is that going to be?
What math?
So this is going to be 25%. Now, how many did you see that were actually heterozygous healthy?
50%.
That'd be half of them. And how many poor souls had the disease? 25. They better add it to 100, right? And now, to make these things answers, I come back over here and I just simply add underlines to them. And then I'll copy this thing. It tells you all the various characteristics. It tells me how many people are going to have sickle cell of my 4. This is describing phenotype, and that's describing genotype. And my Punnett square can tell me all that stuff. If I put this picture over here, I would have this picture for this one, this picture for that one. And I gotta go back and find the other picture for this one. I want to show pictures too, which I might one day do, knowing me. I want to save this, so next time I do it, they'll have those things. All right, now. Don't need that anymore. All that's left